Assalamu alaikum everyone, my name is Saleha and today I'll be giving you guys the lecture titled Physiology of Vision. So this lecture is not too difficult. There is mainly only 4 or 5 concepts that you need to grasp very well, but it appears to be a dense lecture, which is why it's kind of scary. But I've broken it down and inshallah it's going to be very simple and easy to go through. Now what I've done is on a lot of the slides, I either highlighted stuff in red, like I just made the text red, or I bolded certain things and I made it red, or I added like yellow highlighter in certain places. So if you're in a rush and you want to speed through this lecture, you can go through it focusing on the red text and the highlights. And inshallah, that will be enough to grasp the contents of this lecture. So let's begin. First, we're going to talk about the retinal layers and the electrical responses of the different cell types in the retina. Then we're going to move on to the different light sensitivities of the fovea in the optic disc. Then we're going to talk about the transduction process for rods and the different types of cones. Then moving on to the different functional properties of scotopic and photopic vision. So essentially vision in the light versus vision in the dark. And then we're going to talk about receptive fields. That's pretty much it. In the final part of this lecture, we're going to talk about the visual pathway itself, inshallah, and that's going to be it. One more thing before I forget. So when you when you come out of this PowerPoint, you're going to see that on some of the slides, I've listed like definitions on the side over here or over here. I didn't want to fit them on the slide because it was making things very crowded, but it will definitely help to piece everything together, to go over those definitions that I've placed on the sides just by viewing the slides afterwards. All right, now coming to visual light that we are capable of seeing. So the light that enters a human eye that we are able to actually see falls between the wavelengths 380 nanometers to 750 nanometers. Now this is the broad range of lights that we can see. It starts from violet and it ends at red. And you'll notice that the wavelengths increase as we go from violet to blue to green to yellow and then red. Red has the highest wavelength. Red light has the highest wavelength. And lights like blue, colors like blue, purple, these have the lowest wavelengths. Anything that has a wavelength less than 400 nanometers, your eye is not able to detect it. And same goes for anything with a wavelength higher than 750 nanometers. Coming to visual perception. Now, what is visual perception? So visual perception or sight is the ability to interpret the surrounding environment through 4 factors, or essentially there's 3 factors because photopic vision and color vision are more or less the same thing. So photopic vision is the vision that you're seeing during daylight or when there is a high amount of light around you, what you're able to see, your vision depends on your photopic ability. Color vision is also part of photopic vision because you're only able to see color mainly when there's a lot of light. When there's not that much light, our ability to detect color actually decreases. Then there's scotopic vision, which is nighttime vision or vision where light is very limited. And then finally, mesopic vision, which is something in between. So it's called twilight vision. So it's when the light is not very great, very dim, so you can kind of make out colors and shapes, but your eye's ability to detect light is actually enhanced. In scotopic vision, it's very enhanced because that's when you're not able to detect colors at all. And mesopic vision, it's somewhat enhanced because it's like an in-between zone. Now coming to visual perception. So your visual perception depends on 4 things. The first is light sense, which is the ability to perceive light itself and the intensity of light. This is visual acuity, which is essentially the clarity or sharpness of your vision, the ability to see color and the ability to just make out, different colors and things like that. Now coming to form sense is the ability to perceive shapes and, contours of things. Like if I look at a box, how do I know it's a box? That is my form sense telling me that, these are the lines and the edges of this object that I'm looking at. Light sense is what tells me the color. How dark is the box? Is it brown? Is it yellow? Is it dark brown? Is it light brown? That is all under light sense. And form sense is what allows me to determine the contour of things. hold on, let me just turn the pointer on. Okay, there we go. Now coming to the sense of contrast. Now contrast is what allows you to detect how much light is falling on a certain thing. So how illuminated a certain thing is. Picture a red box. If I were to put the red box in the middle of a room, looking at it, you would see the box as different shades of red. Although the whole box is a single shade, because of how the light falls on the box, you see shadows, you see contrast, you see an increase in the depth of the color. That comes under contrast sense. And finally, color sense. This is the ability to detect different colors. So how do you differentiate pink from green? That's color sense. We're not talking about the intensity of the color here. We're talking about differentiating two colors that are completely different altogether. And they both have different wavelengths, as we described on the previous slide. Now, hold on one second. Let me just move this top bar. It's obscuring my view. Okay, that should work. All right. Now, coming to light that we're able to see, light is detected as photons, which is essentially tiny particles of light. And vision is mediated by two things mainly. Actually, no, I don't know why I said mainly. It's just two things, cones and rods. So cones detect your vision in bright light or the daylight, which is photopic vision. Rods detect your vision in scotopic light, so dim light or no light, minimal light. And then when there is something in between, like we mentioned on the previous slide, mesopic vision, that is detected by both rods and cones. Okay, that's all you really need to know from this slide over here. Okay. So a little bit more about the properties of photopic and scotopic vision. There's a phrase called dark adaptation, which is essentially the transition of your retina from being able to see in a lot of light, moving to being able to see in very dim light. Now, naturally, as you can guess, the eye's ability to see color and see objects in very bright light is different from very dim light. So in bright light, we're able to see color, we're able to see contrast, we're able to see contours of shapes and objects more. But when it comes to dim light, you can kind of imagine your body goes into like a protective mode. So in this sense, it's like a primitive ability of your eye to detect any light possible to help you get an understanding of your surroundings. So in bright light, your visual acuity is the greatest. That is when you're able to see colors more, shapes better, things like that. However, when light is minimal, so in darkness, that is when your light sensitivity, the sensitivity of your eye to even the slightest bit of light is the greatest. Another important factor is that, as we mentioned, there is rods and cones. So rods have a pigment called rhodopsin. Rhodopsin is a photosensitive pigment that's present only in rods. It also acts as a G membrane protein receptor. We're going to come to that in a few slides, inshallah. But what happens is when light is very bright, the rhodopsin becomes bleached and is not able to operate properly. And therefore, it makes the rods dormant when light is very bright. Now, on the opposite end of the spectrum, when light is very dim, rhodopsin is no longer bleached, and so it is able to actually allow the rods to be stimulated and to work. Now, certain factors that increase light sensitivity. So in the dark, what are things that happen to your eye in the dark? The first is pupil dilation. Your pupils get bigger. The second is synaptic adaptation of the retinal neurons. And the third is increased rhodopsin, as we just said, because it's no longer bleached. So the rods are more stimulated and better able to detect light. So your light sensitivity is improved. Finally, the transfer of your eye's ability to go from seeing in bright light to seeing in very, very dim light takes about 30 minutes, typically. And that's all you need to know from here. All right, now coming to the distribution of rods and cones in the retina. First, let's talk about the macula. So the macula is a portion of the retina which is present in the center, almost. There's an area of the macula which is called the fovea centralis. Now, what is the fovea centralis? I mean, it says on the slide, but just pretend you can't read it. So the fovea centralis is a portion of the eye where the cones are actually the greatest. One second. So the fovea centralis is a pit in the center of the macula that is composed of cone cells, and it has the highest visual acuity of any region of the retina. Just for further clarification of what the macula is a spot on the lateral side of the optic disc near the center of the posterior wall of the retina. It's an area of the retina which lacks blood vessels, and it also contains the fovea centralis, like we just mentioned. So it's a central portion of the retina, contains the fovea centralis. What does the fovea centralis contain? It contains an abundance of cones. So visual acuity is greatest where? At the fovea centralis, because cones are responsible for visual acuity. Now, as you can guess, rods are not present at the fovea centralis. So where are the rods present? The rods are present right around the fovea centralis. So you can picture this as a fovea centralis with the highest amount of cones. The rods are in the greatest abundance around the fovea, but not actually in it, okay? All right, now let's come to what the purpose of the fovea is. So the fovea centralis has the greatest number of cones, as we just said, and it is where the visual acuity is highest. But let's talk about the features of the fovea centralis. If you look at this diagram here, you can see that the fovea is where the membrane the layers of the eye are the thinnest. This is because this is where light has to penetrate the fewest layers possible, and it ensures perfect light access to the rods and cones, the cones present at that area. Imagine all the light that's entering the eye being focused onto the fovea. It's very important for the fovea to be able to grab in as much of the light as possible. So as you can imagine, the layers here are very minimal to allow this to happen. Now, what is the optic disc? The optic disc is a region of the eye, basically located here. This is the optic disc, and it is where the optic nerve and the optic arteries and vessels, central retinal artery and retinal vein, all those things enter and leave the eye from here. As you can imagine, the optic disc does not have any rods or cones because its function is to serve as the entry point for all the nerves and blood vessels and everything. So there's no rods and cones here. What does that mean for us? That means that this point becomes a blind spot. This part of the eye has no ability to detect light because there's simply no rods and cones. Now, this is a physiological blind spot. However, there are some cases where you can have damage occurring to the retina, like in certain places. Let's say if there was damage here, this would cause a blind spot in that person's eyesight in one of their eyes if the damage is only on one side. This damage can be, on the macula, it can be on the retina or the optic nerve, and it would cause a blind spot. Just for you guys to perceive better, this is the optic disc and it is medial to the fovea. All right, coming to the basic circuitry of the retina. Now there is a 3-neuron chain that we need to know. Before we go on to that, let's look at this diagram here. Light is entering the eye from here, and this is all the retina. Now this layer of cells is mostly transparent, so light is allowed to actually go directly all the way to this epithelium over here, which is the pigment epithelium. Pigment epithelium is very dark, which is what allows the light to reach here and not be reflected off, therefore allowing the rods and cones to detect as much of the light falling on the pigment epithelium as possible. Now, what are we talking about when we say the 3-neuron chain? So it starts with the photoreceptor, which are the rods and the cones. Then it goes to the second neuron, or the second cell involved, which is the bipolar cell. So photoreceptor P, bipolar cell B, and the ganglion cell, which is here, which is the third cell in this neuron chain. Now there's differences in acuity between rods and cones, as we discussed earlier, but let's talk about why there's a difference in the acuity. What happens with the cones is each cone synapses with a single bipolar cell, which then goes on to synapse with a single ganglion cell. The ganglion cell then goes to the optic nerve, and that goes to the brain, and so on and so forth. Now, you can notice here that it's very focused. It serves one purpose and one purpose alone. So you can imagine that any light that hits the cones is very concentrated and it has a very set pathway. Therefore, cones have higher acuity and lower light sensitivity. Now, in contrast, because they have lower light sensitivity, why? Because let's say if light didn't hit this cone and it only hit this cone would have no signal at all. Nothing would be transmitted. So it has minimal sensitivity. The cones have minimal sensitivity. On the other hand, each rod synapses with many rods synapse with a single bipolar cell. So this one synapses with, like, let's say it was synapsing here. Let's say this one was also synapsing to this bipolar cell. That means that if light hits this rod, it would be transmitted. If light hits this rod, it would also be transmitted. This means that the light sensitivity of rods is greater. But on the other hand, it's less focused. It's less, selective. So therefore, visual acuity is lower in the rods. Final important thing on this slide is that glutamate is released when the rods and cones communicate with the bipolar cells. It is the main communicating signal transmitter within the ganglion cells. All right, now coming to light on and light off bipolar cells. There's two types of bipolar cells that we're looking at here. There's light on bipolar cells and there's light off bipolar cells. As we said on the previous slide, glutamate is what is released. So if light shines on a photoreceptor and it reaches a bipolar light on cell, this cell is inhibited by glutamate release in the dark and it is activated in the light. On the other hand, if this was a light off bipolar cell, it would be excited by glutamate release and inhibited by the light. So whether glutamate acts as an excitatory or inhibitory signaling substance, that depends on what bipolar cell it's actually landing on. So essentially what happens here is one stimulus can either create a on signal or an off signal depending on where it lands. And all of this is dependent on a single neurotransmitter, glutamate. Okay, yeah. So the bipolar cells then will either form synapses with either excitatory or inhibitory synapses with the ganglion cells in the visual pathway. All right, another thing. So bipolar cells are capable of transmitting graded potentials, but not action potentials. Now this raises the question, what is a graded potential? A graded potential is a variable strength signal that can be conveyed over small distances, and it can be of different intensities. However, an action potential is a massive depolarization, and it is transmitted over long distances. So think about graded potentials as a tiny signal between two cells that are adjacent to each other. An action potential can be a single, transmission that goes all the way through the length of a neuron. So it's much larger and bigger in magnitude. Okay. Also, graded potentials can vary in amplitude. However, action potentials usually have fixed amplitudes. All right. So ganglion cells are also of two types: on center, off surround cells. Off center, on surround cells. We'll get to this in a few slides, inshallah. We're going to talk about it. So ganglion cells are the first neurons that are capable of transmitting action potentials. So these are the photoreceptors. This is a bipolar cell, which only transmits graded potentials. And then this is the ganglion cell, which transmits action potentials to the optic nerve and to the brain and so on. Now, one more thing is that horizontal cells, which I've circled here, they provide lateral inhibition. We'll come to this in a few slides as well, but for right now, all I need you to know is horizontal cells provide lateral inhibition from the cones receiving light and surrounding to the synapse between cones receiving light in the center and a bipolar cell. Now, retinal pigment epithelium. So this is essentially describing the importance of the retinal pigment. It is because it's very dark, so it absorbs all the light and prevents reflection. What exactly absorbs the light? It's the melanosomes, the pigment present in the melanosomes. All right, our first checkpoint, amazing. So rod synapse with what? Single or multiple bipolar cells? Cells. Rods synapse with, hold on one second. many rods synapse with a single bipolar cell. I think I made a mistake. So rods synapse with single bipolar cells. Yes. 1 second. Hold on. Let me just correct this. Many rods. Okay, I'm back. Sorry. Okay. Here we go. I hope that's clear. All right. So do rods synapse with single or multiple bipolar cells? Many rods synapse with a single bipolar cell. Now, does this allow for greater visual acuity or light sensitivity? This allows for greater light sensitivity, but lower visual acuity. And light sensitivity is highest where? In the darkness. Therefore, rods work best in the darkness. Why? Because they have that one special pigment, which is bleached in the light, but is efficient in the dark and stimulates the rods in the dark. Now, the next one, do cones synapse with a single or multiple bipolar cells? Cones synapse with a single bipolar cell. One cone synapses with a single bipolar cell. This allows for greater visual acuity or light sensitivity. The answer is it allows for greater visual acuity, but lower light sensitivity. Visual acuity is highest in greater light. Therefore, cones work best in the light. All right. Now coming to, just a summary slide of cones and rods with a little more information. Cones are cone-shaped, light sensitive, less sensitive, operate in highlight, and they transmit color vision. As I said earlier, color vision is possible only in the light because that's when your cones operate. But when light is dark, your rods are what's functioning, and they only have black and white vision, monochromatic. So the rods are rod-shaped. People also sometimes describe the cones as flask-shaped cells. So you might see questions which say, they're examining a part of the eye or something, and they found these, and they were shaped like flasks. What are they? Cone cells. Okay. So rods are rod-shaped or more rectangular. They operate at night, and they have monochromatic vision. Okay. So rods and cones are selective light transducers. The dark pigment layer of the retina absorbs stray light and prevents reflection. The discs in the rods and cones are the site of transduction. Discs and cones are pigmented and filter the light of different wavelengths. And this is what allows cones to actually detect different colors. The transduction process is mediated by pigments in the discs. For example, the rods have a certain pigment, rhodopsin. So these are things that we've already covered. Now coming to the transduction process. Hold on. Oops. Okay. So the process of phototransduction is similar for rods and cones. The visual pigment plays a very important role, rhodopsin, and it's composed of two sections. So remember how I said it's a G protein membrane receptor? That's the opsin part. And it's also a vitamin A derivative, and that is the light absorbing portion of the pigment. So when the light is very intense, what happens? Focus here. So when the light is very intense, the rhodopsin becomes bleached, and this decreases cyclic GMP, no sodium influx, and the retina is no longer, this retinal no longer binds to opsin, which was the G protein membrane receptor. But now when there is no light, rhodopsin is not bleached. This allows retinal to bind snugly into the binding site of opsin, the G protein receptor, which allows a greater amount of cyclic GMP, greater sodium influx into the cell, and the cell becomes depolarized. So when light is dim, the rods become depolarized, allowing them to transmit a signal. When the light is greater, the rods become hyperpolarized, so no signal. As simple as that. All right. Oops. So color vision is owed to the light-sensitive visual pigments that are bound to the cell membranes of the photoreceptors in cones primarily. but both rods and cones have these visual pigments. Rods only have one, rhodopsin. Cones have three pigments, which can be excited, selectively for blue, red, or green light. When all the rods for blue, red, and green are activated at once, that is when you get white light. Now, the variations in color are due to the different amounts of red, green, blue in everything that we see. And this is called the trichromatic theory of color vision. All you need to know here. Okay, now for measuring wavelengths. We mentioned this on one of the very first slides, but essentially shorter wavelengths cause the green receptors to fire because green light has a shorter wavelength, something like, I think it's around 450 maybe. And as you get closer to 580 or even more like 600 and 650, that is when red light starts reaching the eye, and that's when the red receptors start to fire. So essentially you have a mix of wavelengths. This mix of wavelengths allows you to detect so many colors. The number of colors that we can detect is nearly infinite. And it all depends on our cones. Fun fact, by the way, there's a type of fish, I think it's a shellfish, maybe, which has 17 different types of cones in its eye. So humans only have 3 cones, red, green, and blue. This fish has 17 different types. Just imagine the colors that it could probably see that we can't see. This always has fascinated me. But yeah, you guys can look it up. I'm not sure if it's a shellfish or a type of snail, but it's like some underwater, deep water fish thing. So yeah. Anyway. Okay. Now coming to what happens if you're missing one type of cone or, one certain color cone. This is when you come to, people with I think it's dichromatopy or I'm not sure how to pronounce it. Anyway, they're called dichromats. What happens is they are missing one certain type of cone in their eye. And that prevents them from being able to see that color. This is not the same as color blindness. So color blindness is an inability to perceive colors altogether, even if you have the right cones. These people are generally missing just a certain cone. So that means that they are relatively colorblind. However, they are able to tell what most colors are because they can kind of make out that, okay, they can't see green, but they can see that this might be like an olive shade of green because they can kind of see the purple hues in it and so on. Monochromats, this is much rarer. It's called monochromacy, I think. Something like that. They have no cones, and this is very rare. But essentially, most people are trichromats, and they can see all 3 colors. One thing that is just helpful to know, so these are things called Ishihara tests. And I'm sure you guys have seen these before, but this is what you use to determine if someone has dichromacy or monochromacy. So someone who has one cone missing, one type of cone missing, they may not be able to differentiate between like blue and green. So this might, probably not this. Let's say this might all kind of look the same, or, maybe this might look the same. And you use Ishihara tests to kind of assess where the visual acuity of these people lies, of these patients lies. Now coming to retinal ganglion cells, I believe we're pretty much halfway done, I hope. All right, so there seem to be 2 types of ganglion cells in the retina. The first are large magnocellular ganglion cells, or M cells. They carry information about movement, location, and depth perception. That's all you need to know. Then the smaller cells are parvocellular ganglion cells, or P cells, and these transmit signals to determine color, form, and texture of things that you see. And that's pretty much all you need to know. Final part is there's an area of the retina which connects to a ganglion cell. That area of the retina that reads from that ganglion cell is known as the receptive field, and that is where activity is recorded from that ganglion cell axon. Okay. Now coming to image formation. The processing of visual information in the retina involves the formation of images. First, you have light that falls on the photoreceptor, the rods and cones. This signal is altered by horizontal cells. Remember we mentioned horizontal cells do lateral inhibition? That's what these cells are important for. We're going to come to that in a second. Then it falls to the bipolar cells. The signal goes to bipolar cells. Here, the signal is altered by amacrine cells. And then finally, the ganglion cells are the third cells in this process. All right, finally, we have reached the receptive field. The receptive field is a region of the retina where the action of light is altered depending on where the light falls. Wherever the light falls, that changes the firing of the neuron. So remember how we mentioned there's on center, off surrounding ganglion cells, and then there's off center, on surrounding ganglion cells. So we're going to break that down right now. So let's talk about on center cells first. When light falls on the center of this region, this will activate the on center, off surrounding cell and cause it to fire rapidly. This ganglion cell to fire rapidly. But let's say if the area of the receptive field where the off center on surrounding cells are located, when light falls on those cells, this causes them to be inhibited. They do not transmit a signal. Now coming to light on the surroundings only. So the cells in the center, not just the center, the cells in this area, because they are being hit by light. Hold on, I think I'm confusing myself. Okay, so the cells in this area are not being hit by light, and they're not transmitting an action potential. On the other hand, the cells in this area are being hit by light, and so they are transmitting because they're on surrounding cells, off center, on surrounding cells. Now, when there's no light hitting either regions, neither of the cells are transmitting an action potential. And when there's light hitting both the regions, the on-center, off-surrounding, the off-center, on-surrounding cells, this is when light is being transmitted, but minimally, because you can imagine both of these are kind of canceling each other out. Let me go over these again. We're going to come across them in the upcoming slides, so hopefully it'll become clearer. So this is when light falls in the center, and this is an on-center, off-surrounding cell. The signal is being transmitted. The action potential is happening over here because this is an on-center, off-surrounding cell. Now, when this area expands, you see how it expanded? The on-center, off-surrounding cells are transmitting even more signals because the light is brighter. There's more of it, so more action potential. Now, when it expands to a region here, which is the off-center, on-surrounding cells, these are the cells that turn off when the light reaches them. And that is why the action potential that you see here is now subdued. It's lesser than it was on the previous slide. Now, if I were to expand this more, it's even lesser now because it's almost like the effects are being, canceled out because light is falling on the positive area on the on-center, off-surrounding cells, and it's being canceled out by the light that's falling on the off-center, on-surrounding cells. Now, if light was not falling here and it was only falling here, then there would be one action potential generated from the on-center, off-surrounding cells. So here the on-center, off-surrounding cells, yeah. So there's nothing reaching here, very minimal, and so there's only one. So this is an example of how the light falling on this area on the receptive field is canceled out, when it's falling on the on-surround, off-center cells, off-surround, on-center cells. So let's look at this. This is actually a very good representation. Center cells here, light is falling over here, strong action potential. Surrounding cells, when the light falls here, they don't have an action potential because they're the complete opposite of these cells. However, there is light when there's no light being, when there's no signal being transmitted here, there is a signal being transmitted there. So essentially it cancels out and it, produces a certain wavelength of light that is transmitted to the brain. Now we're talking about off-center, on-surround cells. So here, when light is falling here, the surrounding cells have a much stronger firing range or firing rate. And the center cells, there's minimal light falling here. So they have a smaller firing rate, and it's, it doesn't take place at the same time. So again, they cancel out, they act in opposite ways. Now, why does this actually happen? So this is when lateral inhibition actually takes place. You'd be like, okay, so why does it matter when light is falling in a certain area? Why does it have to be canceled out by light falling on the opposite area or the adjacent area? This is because lateral inhibition allows you to focus better on the shape of things. The contour of objects and the borders helps you differentiate between, this lateral inhibition helps you differentiate between round things versus sharp things versus, edges and things like that. So diffuse light is ignored by the ganglion cell, but when light is focusing exactly on the center, that would activate a center on surround off cell, and that would generate a rapid firing rate from that cell. But on the other hand, it would cause the adjacent cells to not have a rapid firing rate. That is when the light is focused. So this allows you to see sharper vision, essentially. All right, so what is a receptive field? Area of the retina where the presence of a light stimulus changes depending on the activity of the neuron. So it changes depending on what type of cell light is landing on, the amount of light that it's landing, that there is, how much is being spread out. Is it only on the center? Is it being spread out more? Is it diffuse? Because if it's diffuse, then minimal transmission happens because the two cancel out. But if the light is only on the surrounding cells are going to be transmitting, and the central cells are not going to be transmitting as much. If the light is only on the central cells are going to be transmitting more, and the surrounding cells are not going to be transmitting as much. I hope that's clear, and I hope that wasn't too confusing. I know it's a little up and down, but I hope it makes sense, inshallah. Okay, why are receptive fields important? Because they allow you to see clarity of your vision. They allow you to see shapes and curves and contour, and basically sharpness of your vision is determined by the receptive field. Okay, let's move on. I think we are almost done with 3/4, or maybe I'm not sure actually. Let's see. All right. Now coming to maybe the most important part of this lecture, the visual pathway. Here we're going to discuss the normal pathway, and then we're going to move on to quickly discussing the pathologies of the pathway. I'm not going to spend very long on it because I've included a lot of images, and each image has a lot of description with it, with definitions of, where each pathology happens. So hopefully, inshallah, it's going to be very clear. But you guys can read this PowerPoint later because I've included multiple images to explain it with explanations. So hopefully that will clear it, inshallah. All right, now coming to the visual pathway. Let's move on one slide before we talk about this slide. This is the visual pathway. Now, I want you guys to bring your attention to these two circles here. This is the nasal field. Sorry, this is a nasal field of the right eye. This is a nasal field of the left eye. Now, when you're looking at vision from the left, vision from here, okay, it's going to fall on the nasal side of the left eye, and then the temporal side of the right eye. So all the vision that's coming from here is going to fall here, and it's going to fall here, right? Now, what happens is everything that's being transmitted from the left visual field here eventually is going to go to the right side of the brain. Notice how this green area and this green part, both of them are transmitted through the optic nerve here, where it partially decussates. So what happens is everything that's coming from the left side decussates to go to the right side of the brain, and that's where it's processed. Now, the opposite happens for vision that's being transmitted from the right side of the eye, or the right side of the visual field. So this is, let's pretend this is the right visual field. Everything that comes from here is going to land here, and it's going to land here, right? So it's going to land on the nasal part of the right eye. I'm talking about the part of the eye, not the visual field. So the nasal part is here because like this would be your nose, right? So this is the nasal part of the right eye, and the temporal part of the left eye. This is where your visual field would land if you're looking at stuff coming from the right. Now look at the orange lines. So the orange lines go here, and it goes from here through the optic nerve, past the optic chiasm, and then it decussates. So everything that's coming from the right eye, that's part of the right side of vision, eventually goes to the left side of the brain. Same goes for anything that's coming to the left eye, that's part of the right visual field, that will also go to the left side of the brain. So what do we gather from here? The right brain gets vision from the left side, or the left visual field. Everything that lands from here, everything that comes from here, goes to the right side of the brain. And the left brain receives visual information from the right side. So everything that starts here, all the visual information here lands here and here, and it then goes down like this, and it's transmitted at the left side, at the left brain. Okay. One more thing is partial decussation, as I mentioned earlier. So it's not a full decussation because not the entire optic nerve switches pathways. It's only the part of it that's like relative to this side. So right vision will switch to go to the left side. I mean, sorry, not right vision. Vision from the right eye will switch to go to the left side if it's coming from the right visual field. Vision from the left visual field will switch to go to the right side. Yeah, that's pretty much it. To cover the pathways, so light lands on the retina, on the fovea, eventually reaches the optic nerve, goes through down here, reaches the optic chiasm, partially decussates, eventually goes to the optic tract, and it reaches the lateral geniculate nucleus. Now, vision from the lateral geniculate nucleus actually gets transmitted separately through different striations here. So there's the Meyer's pathway, which we're going to discuss in a little bit. And the Meyer's pathway essentially transmits, it's pretty much here, it transmits visual information from the top of the field. So it's these two fibers or strands that you can see here. Anything that comes from the top corner here, and the top corner here, it will be transmitted through Meyer's pathway. We're going to come to that. So here's another diagram. This describes Meyer's loop properly, shows you the different, radiations, and it shows the dorsal optic radiation. The dorsal optic radiation transmits information from the lower quarter. So imagine this is the lower quarter of vision from the right side, okay? It all goes down here, goes to the left side, then it goes through the dorsal optic radiation. Okay. This is the doctor slide. It just discusses whatever we just talked about. Okay, so this is where we talk about the actual pathologies of the visual pathway. So I need you guys to get creative here and visualize with me. Okay, so let's talk about number one first. If there was a lesion or an injury that cut the optic nerve, because this is the optic nerve here, the optic nerve coming from the left eye, if this was cut right here, what would happen? So every part of the visual field that you're receiving from your left eye, whether that's from the right or it's from the left, it has been cut off completely. So your left eye is visionless, essentially. So you would not be able to see anything from the left eye. This is called monoocular vision loss from a single eye. Okay, now let's go to number two. So let's say if the optic chiasm was damaged in the middle. Let's say there's a lesion or trauma or maybe a tumor that was growing and overgrowing to this part, and it was pressing the optic chiasm. Now what would happen? So what is being compressed here? What's being compressed is the lateral vision that's coming from either eye. So it's not just the right side, it's not just the left side, but it's both, temporal fields of vision from both eyes. So the sides, they're going to be transmitted here. This has to be this would decussate, and the green one would decussate, but because there is a lesion here, it's not able to do that. The vision is not transmitted, and so you get something called bitemporal hemianopia. It's also sometimes called hemianopsia, but more commonly they call it hemianopia now. And this is how you would see it. You would see closing off of the vision on both sides when you try to look laterally. So you would see like darkness in those areas. Why? Because the fibers that are decassating are blocked off. So anything that's coming from the lateral side here, which would land here, anything coming from the lateral side here, which lands here, these fibers are decassating, and these fibers are decassating, and they're blocked off because of the lesion or the tumor, and that results in bitemporal hemianopia. Now the third thing, number 3. This is when you have a lesion in the optic tract. Yeah. Yes. Just making sure. Okay. This is when you have a lesion in the optic tract. Now everything over here is coming from the right eye, except those decassated fibers that are from the left. Now, what happens if there's a lesion here? It means that everything that's coming from the right visual field, which landed here, these fibers then decassated, and everything from here as well landed on this part of the left eye. These fibers did not decassate, but essentially everything that's orange, so coming from the right side of the visual field, landing here, these pathways are completely cut off now. So anything that's on the right side of vision, the eyes are no longer able to see. So you might confuse this with number 1, but it's not the same, because number 1 was, visual pathways from only a single eye. There was no other pathway involved. There was no decassating pathways or decassating fibers. It was just the single, visual information from this eye alone, which is why you get complete loss of vision in one eye only. But when you have number 3, where there's a cut in the optic tract, that means loss of vision from one side entirely, not just one eye. Okay, now coming to Meyer's loop, number 4. Remember how earlier I said that Meyer's loop transmits vision from the top quarter of your visual field, essentially the top half, but each Meyer's loop transmits a quarter. So the left Meyer's loop on this side transmits this top quarter of vision and this top quarter of vision. The Meyer's loop on the right side transmits this top quarter of vision and this top quarter of vision. So together, the Meyer's loops on both sides transmit what? They transmit the top half of your visual field, top half, okay? Now, if there's a lesion affecting the Meyer's loop or Meyer's bundle, the lower bundle, this would affect the vision where? On the top quarter. This is known as contralateral upper homonymous quadrantopia. It's also called a pie in the sky sometimes, because as you can look at this, it literally looks like a slice of pie on the ceiling, or on, in your top visual field. So it's called pie in the sky. Now the opposite happens when the lower fibers are disrupted. So the dorsal optic fibers, these were transmitting information from where? The lower quarter. So the ones on the left are transmitting right visual information, right visual field information from here, and right visual field information from here. Goes down from here, all the way out here, and this way. And a lesion over here would cause a pie on the floor formation, which is called contralateral lower homonymous quadrantopia. All right. Now, sometimes what happens is there's a presentation that's similar to right homonymous hemianopia, except the region of the macula is spared. And this happens when it's a little advanced, but this happens when there's a certain area of the brain that has an infarction maybe, and it just spares these fibers that are coming from the macula. So while one side of the vision is lost, the area of the macula is actually still intact. That vision is intact. Now this is known as, now coming number 7, this is known as homonymous central scotoma. Remember how we mentioned earlier how there's certain blind spots in your eye? There's physiological ones, which is the optic disc, and then there are some which are pathological. So this is an example of a pathological blind spot. But this is, because it's happening in this region of the brain, it involves the right side of the macula from both eyes. Now, if this was, let's say, on the right side here, it would be the opposite. So it would be the left part of the macula from both eyes. Okay, I hope that's clear so far. But again, that part is a little advanced. So 6 and 7 are not as important. What's more important is 1, 2, 3, 4, 5. But you do need to understand the concepts behind 6 and 7. This is again the same thing. Okay. Finally, other associations that are important to know. So optic fibers project to several places besides just the primary visual cortex. There's a suprachiasmatic nucleus of the hypothalamus. This is applicable for circadian rhythm. So what allows you to, sorry, one second. All right, my apologies. Yeah, so the circadian rhythm or circadian rhythm is, how your body regulates sleep, right? So when there's a lot of light coming into your eye, that goes to the suprachiasmatic nucleus of the hypothalamus, prevents it from releasing, those hormones that cause you to go to sleep. But when the light is dim, that eventually transmits to the hypothalamus as well, and your body realizes, okay, it's time to get sleepy now, time to go to sleep. And this essentially is regulation of the circadian cycle. The pretectal nuclei. Now, these are very important as well because these mediate the light reflexes. Now, I'm sure you guys know that when you shine a very bright light into someone's pupil, it's going to cause the pupil to constrict. How does that happen? What is the association between light entering the eye versus the pupillary muscles causing the pupil to constrict? Where is the link? The link is the pretectal nuclei in the midbrain. And it's essentially a reflex. It's a pupillary reflex. Light enters the eye. It is transmitted through the visual pathway to the pretectal nuclei. Sorry, not through the visual pathway. It's a separate pathway. It's transmitted to the pretectal nuclei, which then causes a sympathetic response and causes your pupils to be constricted. The opposite happens in dim light. When light is dim, the same pathway is utilized. The pupillary reflex pathway. It's called the pupillary reflex pathway. And this time, parasympathetic stimulation causes your pupils to become bigger. As simple as that. All right, now the optic fibers also project to the superior colliculus. This controls rapid and directional movement of both eyes. And finally, the ventral lateral genicular nucleus of the thalamus and the basal brain regions. Okay. And oh my God, we're done. So a summary of everything that we've covered so far, or at least most things that we've covered so far. So we discussed what visual perception is. Visual perception is the ability to interpret the surrounding environment through photopic vision, through photopic vision, which includes daytime vision and color vision, scotopic vision, which is dark vision, and mesopic vision, which is twilight vision or medium light vision. The vision is detected through retinal cells rather than cones. It's mediated by cones in bright light, rods in low light, and then both rods and cones at intermediate intensities. Visual acuity is greatest in daylight when the light is greater, and it's least in the scotopic state or in darkness when light sensitivity is maximal. And that's actually it. There's a few slides that I had to hide because I didn't have enough time to go over all of them. That's actually it for this lecture. Just a quick review. Here's another image that describes the visual pathway. This was the image where I added all the, lesions and everything. This is a doctor's original image. And it essentially describes everything that we went over. So pie in the sky, the Myers loop lesion, pie on the floor, which was what was pie on the floor? Yeah, dorsal optic pathway lesion. Optic chiasm lesion is going to cause bitemporal hemianopia, and also the dorsal optic pathway is the same as the parietal radiations, number 4 here. Monoocular loss is when there's a complete cutoff of the optic nerve from one eye, so complete vision loss from a certain eye. And yeah, everything that we've discussed. All right, one more thing, my name and email are in the top of this PowerPoint. If you guys have any questions, please do reach out. I know I covered this lecture quickly. I hope it was enough, inshallah. And I hope that this explanation helped you guys with understanding, inshallah. And all the best in your exam. I know it's very soon. Good luck. And yeah, please feel free to reach out.